I'd like to welcome Robert McLaughlin from the University of Western Australia.
Good afternoon. So my name is Robert McLaughlin from the University of Western Australia. We're talking medical devices. Breast cancer surgeons cut blind. They know where the tumour is, but they don't know where it stops. Every year, 100,000 women in the US will have a tumour removed from their breast. 25,000 of those women will have to go back for a second surgery because the surgeon got it wrong. They left some cancer in the woman. We've made a device that helps the surgeons find the edge of the tumour. It's a miniaturized fibre optic probe encased in a needle, a microscope-in-a-needle. And what's really cool about that is it allows us to do optical biopsy deep below the tissue surface, as deep as you can insert a needle. So the surgeon can actually test the tissue as they're cutting. We have a world-class team for this. I'm a ex-Siemens product manager. I've taken 3 medical devices through to commercial release. Professor David Sampson, head of the lab, has over 20 years' experience in optics and microscopy. Our clinical champion, Professor Christabel Saunders, is one of Australia's leading breast cancer surgeons, and a team of 16 engineers in our lab. For funding, we're looking at starting off with 5 employees. We have an increasing that. We have a well-defined plan of development in the first year, trials in the second year, product refinement in the third year, regulatory approval in year 4, early sales in year 5, and medical device industry, I'd anticipate market traction by year 8. But the critical point there is year 4 regulatory approval, because that's the point at which we intend to be acquired by your St. Jude or Siemens or GE. We have done initial trials on human ex vivo tissue, and we can differentiate cancer from non-cancerous tissue. We have patents protecting aspects of this technology and strategic partnerships with companies like Vitran, where we're looking at how we automate the production of these probes. People like Victor Yang, neurosurgeon and researcher in Toronto. We put one of our probes into a biopsy needle for safer brain biopsy. Caroline Ledoux in Montreal, who provides a key component so that we can make fluorescence microscopes in the needle. How do we monetize this? Disposable probes, 1 needle per patient. It's like selling printers. You don't make money off the printer, you make money off the printer cartridges. We make money off the needles. If we can just halve the re-excision rate, we will save the health insurance companies $1,250 for every patient. Price at half of that, $750. US market size, 75 million annually. In Europe, $120 million annually. We do have competition, intraoperative ultrasound, because this is a lucrative market. However, we believe our probe can see smaller traces of cancer that ultrasound will miss. Now, we're a university research lab. We pull in about $900K per year annually in grant funding. We will spin this out within the next 12 months. We've already had conversations with 3 Australian VCs, and we're working with QSR Solutions on our regulatory strategy. Thank you very much.
OCT IP situation is fragmented, fractured, a lot of people own different pieces. Do you have an indication whether you'll have any IP protection here in the States, or?
Absolutely. So there's, for our patents, the key markets we need to protect are the US and Europe and Japan. Not that there aren't markets in the rest of the world, but if you can protect those, it's not in the interest of the competitor to bother going into it. The basic technology, putting a needle into a probe, that is public domain. What we're protecting is the aspects around it that make that practical. So largely things to improve tissue contrast. What turns it from being a cool thing in a lab into something that's going to be clinically useful. And that's the 3 patents we've got in there.
Besides the disposable, is there electronics or other support systems that are needed, that are specialized for this, that need to be developed?
No, most of the rest of it is actually quite standard OCT, and that's why we're looking at we're good at making needle probes. Other people are going to be better at making OCT scans, so we're looking at partnering for that. Really, our expertise is going to be making those needle probes. And that's also why from the commercialization model, it's the disposable, it's the consumable where we'll make money. The thing I love about needle probes is they're really cheap. They are some fibre, they're a needle and they're a holder. So they really only cost $1 or a couple of dollars to make, but they bring a lot of value. What kills in needle probes is the expertise to actually make them. And that's why our relationships with people like Vitran in looking to automate that process is really what's going to make that profitable. And this should be fully automatable. It's just fibre.
So you've got very good technical collaborators and relationships. What do you have on the medical side where they've shown that this is useful for detecting breast cancer?
So that's certainly someone like Christopher Saunders has been a key part of that. Great thing I love, as an engineer, great thing I love about working with surgeons is they keep engineers real. And one thing I have known is that if fundamentally what you're doing doesn't help them cut someone open better, they've got no time for you. And people like Christopher are excellent for that. The two major hospitals in our city, we work with them closely, we do trials in both of them. We're just starting out. One of the interesting things about the work we're doing in Toronto was that was our first time where we've sent needle probes away to a remote site and getting them to actually use them. That's a model we're starting to explore this year to bring in more clinical collaborators. Something that I've experienced in my past life in Siemens in the US is that surgeons are very conservative. Surgeons don't want to be the first person to do that. And that makes sense because if a surgeon does something wrong, people get hurt. So really those initial trials and who you're partnering with and who are the first surgeons is incredibly critical. Other interesting thing about this is particularly the US, it's very coast-based. So if you're trying to sell to someone on the East Coast and you've got clinical sites on the West Coast, they've never even heard of that place. And similarly with the West Coast. So it really is about identifying those sites early on.
So finding the right clinical collaborators to get those key publications that show that this is effective.
And typically that's going to be your research surgeons. Surgeons just in private practice, it's not they're not interested, they just don't have time. It's surgeons with a research bent who will be open to that technology and also who can then do high profile work.
I'm interested in your statements around augmenting the contrast and so forth. How applicable is the base technology to other types of detections beyond cancer or what have you?
There's a whole lot of areas we do want to go into. Breast cancer makes a lot of sense because it's very good organ to work with, but that's also why we've been extending into brain biopsies. What I have realised is that a single lab or a small spin-out company can't do lots of different areas at once, and so it's much better to choose one, focus on that, and do that incredibly well. What you're saying about contrast, I might point out, I don't want anyone here to leave here before 6 because I think we've got drinks from 5 till 6 and so you should enjoy the rest of that. But if you do happen to leave at 5:30 in the main OCT track, there's an excellent talk. We have recently made an OCT needle probe which will do simultaneous OCT and fluorescence. And we've managed to get the fluorescence sensitivity down to a point that we can see fluorescently labeled antibodies. We demonstrate that on some liver tumor. And once you can see antibodies for me equals identifiers of cancer. So there's a lot of really exciting stuff happening right now in terms of how we can improve that OCT contrast.
Could you comment on how easy it is to use compared to the ultrasonic techniques? I mean, it seems like, is it a small field of view? Does it have a wide dynamic range?
The small field of view is a key thing. This is one of the problems that I have with optical technologies in general, and certainly OCT. You can realistically only see, we always say 2 to 3 millimetres in the literature. I think most researchers don't quite believe that. It's 1 to 2 millimetres on a good day. So you've got to choose your clinical application. The great thing about this, so we're doing this in surgery. The surgeon has decided they're going to cut here. They don't need to check anywhere else. The only question the surgeon has got is there any cancer along this line? Which is why what we're looking at is take your needle probe, stab there, there's cancer there. Okay, I need to change my cut a little bit. So it's very, very focused. The other sort of get out of jail free card here is we don't have to catch all the cancer. That breast will still go for histology, and we're not going to get all of it. But if we can improve their decisions, that saves a lot of money. Right now they get it wrong 1 in 4 times. If we can halve that so that the pathologists less often say, guys, how can you miss some cancer? Then that's something that makes a big difference.
All right, let's make that the last question.
Thank you very much.
For the $10,000 and the $5,000 Edmund, and this is one of those things where I could have had a real bad conflict of interest because I was just there. Microscope-in-a-needle, Robert McLaughlin.
Great job, really well done.
And the other thing I'll say is that even though Perth, Australia is the furthest place on the planet away from where I live, and I'd never heard of Western Australia University before I went to visit. They're 100 years old. They're considered one of the top 100 universities in the world. And in their mission statement, they are seek diversity. And in their very, very, very first admitting class in 1910, was it? 1910, 1/3 of their admitting class was female. Okay, and so it's a very interesting, very good visit. So.
I'd also say, not only do we have the same weather as San Diego down there, and incredible beaches, but actually the university is on a river and we occasionally get dolphins. So definitely consider coming down.
Yeah. So we have one more. Oh, huh?
Yeah.
Oh, we did.
Yeah, Robert?
Oh, Robert, the Edmond optics. So that was 10,000. Here's 5,000. 5,000 more. So you can come in here.